That question being, you got 100 trillion cells. How come it's not 100 million and they're just bigger? How come a blue whale isn't the size of a blue whale because it's got bigger cells? It just has more of them. How come cells are so dang small? And what that worksheet from Friday with the Snowman Lab is basically walking you through is the math of three-dimensional objects, which is basically to do with the fact that as a 3D thing gets bigger, well, you tell me, as a 3D thing gets bigger, what? What's the basic geometric principle here?
Its volume increases.
Its volume increases. She said the ratio between volume and surface area, but it's really surface area to volume. Because if you say it backwards, then it's the opposite, right? So surface area to volume, that ratio gets smaller. And the way to say it that starts with what he said is, as a thing gets bigger, its volume increases faster than its surface area. Volume gets bigger faster than surface area, which means if you go surface area to volume ratio, that number gets smaller. It's a little bit counterintuitive. That's why we spend so much time on this. That's why we're doing like a class period and a half here on this math. It's not hard math. It's just hard to visualize something, a number, any number, getting smaller as a thing gets bigger. But that's what surface area to volume ratio does. And that's important because it turns out the surface area of a cell, you told me my surface area as a person is skin. Well, the surface area of a cell is its cell membrane, right? And that cell membrane is critical to get stuff in and out of a cell that it needs to survive. Oxygen, water, carbon dioxide, all that stuff. Got to get in and out. And the way it gets in and out, this is not an active transport situation, not even mostly a facilitated diffusion situation. It's a diffusion all by itself. It's like that stuff has to diffuse in and out of the cell naturally. Enough oxygen as it gets dropped off in you by your bloodstream, right? By your hemoglobin and your red blood cells. That oxygen gets dropped off outside every cell in your body. It has to diffuse inside every cell in your body. Make it to the mitochondria to complete the electron transport chain. Remember? Turns out, because bigger cells would have a smaller surface area to volume ratio, which you can now think of as, instead of just surface area to volume ratio, you can think of it as cell membrane to cytoplasm ratio. That cell gets too big, it's got less cell membrane per amount of cytoplasm that cell membrane has to support. The cell membrane is supporting the cytoplasm, keeping it alive by allowing enough oxygen in, CO2 out, water in and out. And so basically, big cells don't have enough cell membrane to keep their cytoplasm alive. That is why cells have never, in the 3.7 billion years that they've been around, gotten big. It grows, that's a great question. Yeah, it's not like the membrane can just stretch. It really can't. Because remember, it's made of all those phospholipids in that lipid bilayer. You know, and so pulling them apart is just, that's going to pop the bubble. Right? And so in order to get bigger, it would also have to make more cell membrane parts. Yep. That principle, by the way, that mathematical principle is that any three-dimensional thing, as it gets bigger, its surface area to volume ratio gets smaller. It comes up again and again in real life. There are some things that are explained by that in real life that aren't about cells. And so watch out for brain teasers coming your way that might have that as an answer. Having a high surface area to volume ratio isn't always a good thing. There are times when that's a disadvantage, which would be a reason that something might be big instead of small. Good, so that was that concept. It's kind of a standalone that, explains why we have to keep dividing cells and dividing cells and dividing cells. Your cells just don't keep getting bigger and bigger. They got to divide to make more of you. Good? All right, we have made it to the last thing in the chapter. Because clearly there is very precise control over cell division. The fact that you can get a cut on your arm and it heals perfectly, give or take a little scar tissue. Mostly not. Mostly you can't even tell. The number of times I've screwed up in the kitchen and cut myself is almost infinite, and yet I only got a couple scars. Pretty much cracked my head open in a bike wreck, and yet that's all I got right there. Pretty precise control. So here are a couple of things we know about the regulation of cell division. I say a couple of things because, first of all, we don't want to spend a week on this, but second of all, there's only so many things we know. This is actually a process where the regulation is not entirely known to us. We'll talk about that in a minute. First of all, there are what we call internal regulators of cell division. That is, things that are basically keeping the inside of a cell to make sure that it doesn't divide until it's ready. One example of a family of internal regulators that we understand fairly well are called cyclins. Cyclins are proteins that basically, when enough of them build up, they allow the cell to go into the next phase of the cell cycle. Which we should probably go back. I don't think I ever showed you the checkpoints in the cell cycle. You remember the cell cycle, right? And by the way, from your homework, I mentioned to a couple of you, and also from the quiz, if I do ask questions about the cell cycle, understand that I'm not asking about the individual phases of mitosis. I'm talking about that pie chart that has the G1, the S, and the G2. So let's look at that again real quick. And it turns out there are checkpoints. And these checkpoints happen at the end of G1, the end of G2, and the end of mitosis. Go ahead and draw arrows to those spots on the diagram you made of cell cycle. End of G2, end of M, end of G1. Those are spots where the cell can kind of just stall. Not a bad thing, necessarily. The cell has a job to do. It's going to stick at one of those spots and go about its work. So internal regulators like cyclins, and then external regulators, which are the things that come from outside the cell and respond to events outside the cell, like, for example, adolescence. Stages of life where more or less cell division needs to happen. And this has less to do with checkpoints and more to do with the speed at which a cell is spinning through that cell cycle, speeding up or slowing down the cell cycle. And one example of an external regulator that might be familiar to you, purely because it has been used as a performance-enhancing substance, HGH, or human growth hormone, which is a growth factor among many that the body uses. Okay, and so I think I'll leave for tomorrow the big question here, which is what happens when control of mitosis, which is normally so precise, is lost. As promised, sample essays, you'll be able to do all of them except the last one. The last one will be tomorrow's lecture. These are due on Wednesday, which is also chapter 10 test day. All right, so those are due on Wednesday. Go ahead and use this time to get a good start. We got 10 minutes, and I'll leave you alone.